Thanks for a great kickoff, Mark. So next up, we're going to hear from Mace Rothenberg at Pfizer to give us his view, or maybe more broadly, the industry's view of seamless cancer drug development.
Thanks, Rich, and thanks to all of you for being here. I think this is going to be a terrific discussion. As Rich was presenting the overview and talked about the 4 sections today, I realized that I actually touch on all 4 sections during my presentation. It's the overall perspective. There are some case studies in here. It's integration of what we've learned at any individual stage of development into the next stage. And then it's also the continuous learning. So with that, so we'll start from the high level, what we view as the challenges to seamless drug development in industry. So first we have to ask ourselves, what kind of signal are we looking for to accelerate the development of a new cancer medicine? How strong does that signal have to be? How certain do we have to be that that signal is real? And how big is our experience before we can make a decision? If a surrogate signal is being used, and we really rely on surrogate signals early in development, how certain must we be that the surrogate signal will translate into clear clinical benefit? A question we're often asked by regulatory authorities. How confident are we that the right patient population has been defined for accelerated development, a point that Mark made in his presentation? What investment do we have to make now in order to ensure that the medicine can complete its journey to the market in a rapid fashion? Something that when I was in academia I didn't really appreciate, but we have to begin planning on all of the manufacturing to meet the criteria for a commercial product years before it's going to reach the marketplace. And that's oftentimes before we have positive pivotal data. So we're taking a calculated risk, and if we're wrong, we've spent millions of dollars on a drug that will never see it to the market. If we're right, we've been able to accelerate that rather than waiting to make that commitment until we have positive phase 3 data. And operationally, in industry, the same group may not be responsible for preclinical, early clinical, and late clinical, registrational, and lifecycle development for a single medicine. There are handoffs. So how can we ensure that we coordinate these efforts across groups? So let me break these down into 3 different areas and dive a little bit deeper into these. Signal detection, kind of signal, you're all very familiar with these, and it seems like with the advent of new platforms of therapies, these are becoming even more complex. So it's not just do we use PFS or OS, but now we have spider plots, we have overall survival rates at milestone analyses, as Gidon Blumenthal has shown. We have overall survival, but also have restricted mean survival times when proportional hazards assumption is not met, elevated tail of the curve. So what constitutes a signal? And how do we make sure that we're really keying on the right signal to make our decision? How strong is that signal? What is the context of benefit achieved from available therapies? So what is the unmet medical need? The context of potential competitor compounds in this space, it's not just the same mechanism of action, but it's actually compounds with different mechanisms of action that are actually vying for that same patient population. And then how confident are we in that signal? As Mark said, this could be a chance occurrence, especially if you rely on unreliable data or unstable data. Is it related to patient selection? Do you simply have a good prognostic group of patients that's given you that strong single-arm activity, or is it really something? And how can you determine that prognostic from predictive element that you're looking for? Is it similar to what could be achieved by existing therapies? And how many patients have been treated? What has been the depth of response in those patients? Because if you have a few patients with outstanding responses, that can actually be even more convincing than more patients with somewhat equivocal responses. What about the durability of the response achieved, or disease control, which shouldn't be overlooked either? Has there been meaningful symptom control or relief through patient-reported outcomes? And what's the safety? The next big category is investing at risk and the opportunity costs. The decision to accelerate drug development is not taken lightly. It requires deployment of human and financial resources. It's a multi-year commitment. You can't decide to embark upon a large development plan and then stop it next year if something happens. It has lots of trailing costs that go well into the future. What's the risk tolerance of the organization? And that may change within a single organization over time. And what are the opportunity costs? In other words, if you do this, what is it you're deciding not to do? And how do you balance those two? And the evidence that exists for each opportunity. Governance. It may involve multiple layers and committees within an organization, and the opportunity must be understood by leaders who may not be well versed in oncology or deviating from the phased approach that Mark said we really should put in the rearview mirror. And the timing of the request may be out of sync with the yearly operating plan. How do we make sure that we can move quickly when the signal arises, not just waiting for the next year? Oncology is moving too fast for us to delay in that way. So let me start now by giving one of two examples. And this is a CT PET scan. And you can see the baseline scan is on the left, and the follow-up scan is day 14, 2 weeks after initiation of treatment in this patient. And you can see the extensive liver and nodal metastases and lung metastases in this patient have substantially decreased in just a matter of days. And this is one of the early patients who responded to crizotinib with ALK-positive non-small cell lung cancer. So I think that this, you can say, met our criteria for a very strong signal early on. But let me now step you through the process that we went through in the organization. Crizotinib entered clinical testing before I had anything to do with it, so I don't take any credit for this. It entered in 2006, and its kinase inhibitory profile was well known, and the target was c-Met. It was a molecule and signaling pathway that is felt to be mutated and activated in a very large proportion of patients who had received targeted therapies and then become resistant. So there was a lot of good preclinical rationale. And in phase 1, there were 3 observed responses in 37 patients, which is not at all unusual. This is not what constitutes a strong signal, because when you look across phase 1 experience, the objective response rate in unselected patients is between 5 and 10% historically. But something happened, and that was an observation made completely independent of our company in Japan about the discovery of a translocation between EML4 and ALK genes that actually was causative of lung cancer in mouse models. So we knew that in addition to this being an inhibitor of c-Met, it was also an inhibitor of ALK. So what we did was we actually worked with Academia, Mass General John Iafredi, and actually had him probe the 3 responders, and all 3 of them had translocations of the ALK gene. So that's when we incorporated this new knowledge into changing the direction from this being developed for a c-Met indication to an ALK indication. So first we engaged with the FDA and had to find a reliable companion diagnostic that could be eventually a marketed companion diagnostic, and that took a lot of effort. And then we expanded the ALK experience in the phase 1, so it went beyond just the typical 10, 12 patients you normally treat at the MTD, but we opened it up more broadly. And following advice from FDA, EMA, and others, we opened a second phase 2 trial. The idea was this just a chance of observation in a small number of patients, or can you identify these patients prospectively and see a high response rate of deep, meaningful duration responses? And then, in addition, while this was being initiated, and again in conjunction with the FDA, we initiated not 1 but 2 phase 3 trials. One in first line versus combination chemotherapy, one in second line non-small cell lung cancer against single agent chemotherapy. Now, at the same time, in this phase 1, we also noted that there was a 3rd gene that had been identified in this time that was a translocation of the ROS1 gene. And so we actually amended the phase 1 trial yet again for a cohort specifically looking at these kinds of patients. So we completed accrual in 2011. And this was the response we saw in a ROS1 patient. And you could see in the patient's left lung, that's not fluid, that's actually tumor, because you could see air bronchograms through that, and you could see after 3 months this patient had a virtually normal chest x-ray. So again, very remarkable response. And this shows you the kinase and showed you why we were looking at these three genes in particular. So this is where it stood by 2011. So now, just in the interest of being able to fit everything on a slide, animated it. And so in 2011, after completion of accrual of the phase 3 trial, which was very important for us and for the regulators, crizotinib was approved by the FDA. 14 months later, only after the phase 3 data from the first trial in second-line non-small cell lung cancer became available, did the EMA feel comfortable approving it at that time. And then, after seeing more responses in our phase 1 cohort for ROS1 non-small cell lung cancer, we initiated not one but several separate phase 2 studies focusing on ROS1 positive patients. Now, in the midst of all this, this relates to the issue of incorporating new knowledge as you go along. It's interesting how sometimes a circle comes back on itself. Because remember, this entered clinical testing in 2016 as a c-MET inhibitor. What turned out in the ensuing 8 years was that, number one, amplification of c-MET to the level that we believed was necessary to render sensitivity to crizotinib was a very rare event. But it wasn't until 2014 that a new molecular alteration, not just overexpression or amplification, but an exon 14 skipping mutation was defined, again, in an independent laboratory, that seemed to be associated with driving tumor through this pathway. And in fact, we recognized that this was now an opportunity, and some patients were entered with an exon 14 skipping mutation and had a remarkable response. So now, the third color indicating a third indication, we now amended the phase 1 study yet again to enroll c-MET exon 14 mutation patients in non-small cell lung cancer. And then coming up to 2016, crizotinib was approved in ROS1 non-small cell lung cancer patients by FDA and EMA, not on the basis of phase 3 data, but just on the basis of phase 2 data, not 14 months apart from one another, but 6 months apart from one another. So I think you could see the evolution of the environment, not just how we approach this and incorporated new knowledge, but also how regulators viewed these data and were able to learn from this. And despite some early discussions with the EMA where they hinted that they may want a phase 3 trial to demonstrate that the activity in ROS1 altered patients was the same as the level of activity we'd seen in ALK positive patients, we're able to show through multiple positive phase 2 studies showing very strong signals of deep responses lasting for a long period of time, that a single arm trial and multiple single arm trials was sufficient for approval. And we received that just a few months ago. So lessons learned from this experience: strong signals of activity can be observed in phase 1. Understanding the underlying biology and genomics can alter and enhance confidence in radically shifting clinical development strategy. Expansion cohorts in phase 1 trials can provide unique opportunities to rapidly and efficiently test clinical hypotheses. And Profile 1001, our phase 1 study, has undergone 25 amendments in 9 years and remains open to accrual today. Phase 1, 2, and 3 trials can coexist under the right circumstances, as you saw in our experience with crizotinib. When strong signals of efficacy are detected and confirmed, regulatory agencies are demonstrating increased willingness to act without phase 3 data. And lastly, the end result is that a new agent with truly transformational levels of activity can reach the market years ahead of schedule. So the second example is with avelumab, and Eric Rubin is going to be presenting in the next session his experience with pembrolizumab in the PD-1 space. So certainly this will not be done in any extent of detail like the previous one, but more to give certain lessons and principles for accelerating the development of a very different kind of agent in a very different setting. So this is, in 2013, avelumab entered clinical testing. It is a PD-L1 inhibitor, and I think current estimates are there are more than 20 checkpoint inhibitors in clinical development now, and avelumab was right in the middle of that pack. So what happened was it was able to benefit from the information that was already being generated in this field we knew something about its single-agent activity, we knew something about the diseases that were sensitive, we knew something about potential patient selection biomarkers in PD-L1 expression. So the approach that was taken before we were involved, this is Merck Serono's compound. They opened a phase 1 trial, not just assuming it was going to be, the 20, 30 patient phase 1 dose escalation trial, but actually knowing that it was going to be moving in multiple expansion cohorts. So this entered phase 1 testing in a very different way than what I described with crizotinib. They knew they were going to have a large one with multiple cohorts and learning throughout this. Now, from this, there are multiple directions it can go, and I'd like to just follow up on each one of these and really discuss the approach that's taken. So, scenario 1, in situations where you have an expansion cohort that shows a very strong signal with single agent, you have both efficacy and safety, and you have adequate experience in a sufficient number of patients, and there is no standard of care or an inadequate standard of care, an unmet medical need, this could be the basis for moving forward to registration. In fact, this has been our experience with avelumab in Merkel cell carcinoma, which is currently under review by both EMA and FDA based on the expansion cohort from this study. Now, scenario 2, where you go not just to registration after your expansion cohort, but you go to a phase 2 study. This is where it is appropriate, where a signal is observed with single agent in phase 1, but needs confirmation or refinement. Do you have the right patient population? Do you have the right biomarker? Do you even have the right dose or schedule? And then the phase 2 trial could focus on a specific biomarker selected group of patients, and based on the signal that's seen, that could be the basis for registration. Or if there's a sense that this is an improvement, but it's not clearly a transformational improvement, it can go to a phase 3 trial. Scenario 3, that's where instead of moving forward with single agent, you move forward into a combination of phase 1b. But you do it in a way where the phase 1b is really the preamble, if you will, to a phase 3 trial. This gets to Mark's point of the alpha, beta, gamma approach, a single study, but actually looking at 2 different settings or 2 different parts of that. So you have a strong rationale for the combination. So you have phase 1 data and the single agent, and you have single agent activity for each component. There's a low probability of drug-drug interaction or synergistic toxicity, because when you design one of these trials, you don't open it up at 3 or 4 or 10 sites as you would for a normal phase 1. You open it up at dozens or hundreds of sites because you have to prepare for rapid movement into phase 3. That's a huge upfront investment. So you have to be pretty certain that you're not going to be thrown by what you see in the phase 1b. And a good example of this is what's going on now with both avelumab and pembrolizumab being combined with axitinib in metastatic renal cell carcinoma. Small molecule versus a monoclonal antibody. There's single agent activity with each component and very little overlapping toxicity. And in fact, both of these have been moved very quickly in this model, phase 1b going to a phase 3. And the last one is a phase 1b study where you're really looking at exploratory combinations, where there might be some safety concerns or patient selection criteria that warrant you to go a little bit more cautiously. Do the phase 1b, understand it, refine it, and then when you're certain, then make the same decision as you saw in the earlier slide. Do you go on to a phase 2 or a phase 3 study? And an example of this are IO-IO combinations, where the concern is you might overactivate the immune system, you might see some overlapping toxicities that could be problematic. So I think that this is a good example, and you'll hear more with Eric, because I think with pembrolizumab, they pursued all of these strategies as well. So lessons learned from this experience is that phase 1 trials can be designed to be large and include multiple expansion cohorts, provided that there's ample and compelling reasons for that confidence. And the FDA should be aware, and Janet Woodcock may be commenting on this, is that the FDA has taken steps to limit the size of expansion cohorts. So you can't just say, we'll put on additional patients until we feel we've gotten confidence. but you have to prospectively say how many patients you're going to put on and what level of activity you have to see in order for the FDA to approve that amendment. Multiple paths may be pursued following phase 1 expansion, including formal phase 2 evaluation of the single agent, phase 1b testing of the combination, an integrated phase 1b, phase 3 trial, or moving directly to registration. And finally, significant resources must be committed for a long period of time. This approach is not suitable for most new agents. But successful application of this strategy can shorten clinical development substantially and speed new medicines to the marketplace. So with that, I'd like to thank you for your attention and be happy to have discussion. Thank you.